All right, so we're going to pick up with what will be our last section on viruses by starting with influenza. And so, influenza, again, is one of those diseases that's been with humans probably since as long as there have been humans. And it's just something that is kind of always going to be with us um, because it does such a good job in infecting us. And this flu virus, or flu viruses more accurately, um, cause a wide range of damage from almost little in the way of symptoms until extremely lethal in some cases. So, the most famous outbreak of influenza occurred back in the span between 1918 and 1919, so right around the end of World War One, when between 20 and 50 million people were killed worldwide by the flu. Now, back then, and I mentioned this in a previous section, 20 million people is a lot of people anytime. But back during that time, there were only like 2 billion people on the planet. So you're talking about a huge percentage of the world's population was wiped out in a single year by influenza. Flu viruses are enveloped, so we talked about that before, a way to escape the host's immune system, at least detection, and therefore prevent attack by the immune system. There are different types of flu virus. Type A is the one that's serious. This is the one that has caused all the major outbreaks of flu throughout our history. Type B and Type C are the ones that make you sick, but not really, so much in a dangerous way. It might make you feel kind of crummy for a few days, but after that, you get better and you're fine. Type A, on the other hand, often comes with serious complications and can lead to death, especially in older patients. Now, the way flu viruses are labeled is based on their protein spikes. So there is an H spike and there's an N spike on the outside of these flu viruses. So H stands for hemagglutinin, and this is a protein that lets the virus, or helps the virus, get into a host cell. Neuraminidase is a protein that helps the virus get out of a host cell. So depending on the types of those proteins present on a virus, that's how we label these viruses. So for example, if we look at H1N1, which was kind of a big deal just a few years ago, the so-called swine flu was H1N1, as by the way was the Spanish flu, the one up top here, H1N1 as well. That just means it had the first hemagglutinin protein type and the first neuraminidase protein type as well. Now, these H and N molecules build up mutations, and so this is the reason why if you get a flu shot, which I recommend getting a flu shot. I get a flu shot every year, so I'm not a physician, not a medical doctor, but just from my own perspective, my own point of view, I think a flu shot's worth it. I've gotten one every year for about the past 10 years. And I don't think I've had the flu in those 10 years. I'm pretty sure I actually have not had the flu in those 10 years. So from my perspective, personally, my own thinking is that there's really no reason to not get one. There's only an upside to it, and so therefore I get one personally, but that's just me. The reason why we have to get flu shots every year is because the flu virus changes every year, and because of that, the vaccine that was given last year might not be effective this year against the flu virus, and that's a kind of an ever-evolving sort of situation. This battle between humans trying to come up with a vaccine that will be effective and viruses which are not intentionally coming up with mutations, but just sort of doing so because of how they work. There's this kind of constant battle back and forth between the virus and us trying to prevent the virus from causing damage. The other thing that these viruses can do is they can do a sort of genetic recombination, where if you get two different virus strains in the same cell, they can actually recombine and create a new subtype, which can be especially dangerous because, as we've talked about before, if an immune system doesn't have any experience at all with a particular pathogen, and that pathogen's serious, a lot of times the pathogen can do irreversible damage before the immune system ever has a chance to respond to it. Now, there's also something which is called an antigenic shift, which is kind of related to that recombination I talked about a second ago, but especially when we get these new strains popping up, whether they're new strains from other species popping into humans, whether they're old strains of human viruses that are recombining to create a new strain, whatever the case, these are the situations that have been especially problematic in terms of killing large numbers of people, like H1N1 back in 1918, the Asian flu of 1957, which was H2N2, also type A. This one had over 100,000 Americans killed off in a single year, which again, in 1957, there weren't nearly as many of us as there are now. But, if we look at the COVID-19 outbreak, we don't really know at this point in time, as I'm recording this, how many people will eventually die as a result of this. But it's starting to creep up there a little bit. So, I don't think it's going to get to that level of 1957, but it's definitely a serious pandemic. 1968, there was an outbreak of type A H3N2. 50 million people in the U.S. had it. 70,000 were killed by it. So, all of these are, relatively recent, almost within the past 100 years, except for the Spanish flu. Outbreaks of flu that have killed off hundreds or millions of people, even in this country, much less worldwide. So this happens, and influenza is a very, very real threat. It's not something to be taken lightly, even though it's an old threat. It's one that's not really as shiny and new as like COVID is, or Ebola, or yellow fever. It's been around. People know about it. It's not exciting, but it still kills huge numbers of people every year. So it's one that we definitely should not forget about. A lot of times these viruses do come from places like China as a result of several things, social factors. A lot of times in rural areas, especially, people will live in close proximity to their livestock, birds especially. They might live just outside the home or sometimes even in the home. And so if those livestock are sick, and whatever's making them sick somehow has a shift which makes them able to infect humans, then we get this new jumping into human sort of a thing. And again, the human immune system has never seen it before, so it's unable to fight it off, especially when you get that genetic recombination. The Hong Kong flu, for example, was a recombination between a duck influenza strain and a human influenza strain that created a brand new one which had never been seen by the human body before. Now, emerging viruses are those that are showing up for the first time in people, and so this happens as a result of a couple things. Number one would be strains of flu that jump into humans from other species, which can happen, even in terms of livestock, but especially because humans are expanding where they live into new parts of the environment that have never been colonized by humans before, and so they're running into either strains of viruses or brand new viruses that our immune system has never dealt with before. So it's very, very difficult for our body to fight it off, especially if it's one that does damage quickly and is fatal quickly. Another difficulty with this, and the COVID outbreak has been a good example of this, is how quickly these things can spread once we have things like rapid travel, air travel especially, right? So all it takes is one person to jump on a plane that's sick and travel back to wherever they came from, and then they're going to start spreading it like crazy. One person gives it to 5 people, and those 5 people each give it to 5 people, and it spreads very, very quickly. It only takes one person to start a pandemic, really. Hantavirus is one that is, it's been around for a few decades now, maybe, 30 years or so, 40 years, especially out in the southwestern U.S. If someone picks up hantavirus, it can cause a very nasty type of pneumonia pretty quickly, but it's very, very uncommon to get this. It's actually carried in the waste of deer mice. Deer mice are everywhere. They live here. They probably live in your house from time to time. But at times, these deer mice can carry a virus, which if you inhale large amounts of it can make you very, very sick, especially if you're in a small enclosed space. Now, most of the time, that's not going to be the case. You might have one mouse every now and then living in your house, but that's not even close to enough to cause you to have problems. What we're talking about here is somebody who maybe goes to like a hunting camp once a year, small space. It's been closed up for a year. It's been colonized by mice. They're defecating and urinating everywhere inside that space. And then a person goes there and opens it up during hunting season. It goes in and they breathe in just massive amounts of virus propagules, and that makes them very, very sick. So it's not very common, but it is one that if you're exposed to it can make you very sick if you're not treated. Ebola, of course, we've talked about that before as well. Ebola causes a really, really nasty hemorrhagic fever, which is fatal if left untreated, but otherwise it is somewhat treatable, especially if you have access to medical care. With Ebola, the mortality rate is up to 90%. It's not always 90%, but it's up to 90%. That's the number you always see reported. 90% is people who get it and basically don't get medical care for it. That's like your worst case scenario. Otherwise, it's still 60 or 70%, which is huge, but not always necessarily 90%. And the saving grace of Ebola from our perspective as humans is that it's not that easy to spread it if you take proper medical precautions, and for that reason it's not been one that's caused huge amounts of worldwide damage. If you're looking at the number of people that have died from Ebola ever in human history, it's probably 15 or 16,000 total ever, which is a lot of people, granted, but it's nowhere near like the Spanish flu outbreak or even the COVID outbreak for that matter. It's nowhere near as much as some of the other big viral outbreaks because it doesn't spread all that easily. SARS we talked about before as well. SARS was picked up back in the early 2000s by a person who was in close proximity to a civet, which is a type of, it's like a small, the closest thing I can relate it to that we know, like familiar to us here, is like a bobcat kind of. It's a small cat that somebody had in close proximity to them, and it actually was sick, and it actually passed on a virus that was able to colonize the people, which then spread. Caused a lot of panic back in the early 2000s, but never really amounted to very much. It was controlled relatively easily. Mutation rate was low. There's also a vaccine that was being developed, so it's been one that's fairly controllable as well. Now, something interesting that we haven't talked about yet is the role that viruses play when it comes to causing cancer. And so the way this works is that, especially with viruses that are in a lysogenic type cycle, so just to review for a second, lysogenic means that they invade a host cell, they inject the DNA, which then becomes part of the host cell DNA, and then that host cell from that point on keeps producing viral DNA whenever it replicates its own DNA. The problem with that is that the viruses are not especially specific, or I guess specific about is a good way to say it, about where they put that DNA once they add it to our genome. So the possibility is when a virus invades a human cell and injects its DNA, maybe that DNA disrupts a proto-oncogene, for example, like we talked about back in our chapter on mitosis. And so if that occurs, if a gene that normally is regulatory or normally affects the production of, say, growth factors is mutated as a result of a viral gene or genome being introduced, then the cell that has that altered gene could start producing a more rapid rate of cell division. And so that's a really common case, we think, with viruses inducing cancer is by the activation of proto-oncogenes into oncogenes. Remember, proto-oncogenes are genes that have other functions in our genome. They're not bad. They're actually something we need. But when they're altered, they can cause an out-of-control cell division, which is what we call cancer. So they essentially disrupt the cell cycle, and that causes cells to begin to grow out of control. So the most famous example of this, the best documented case of viruses causing cancer, has to do with the human papillomavirus, or HPV. So back in 2006, there was a new vaccine developed by the FDA, or not developed, it was approved by the FDA. Somebody else developed it, but they approved it for use, which is able to prevent infection by HPV. So HPV is a virus or it's a group of viruses that can cause essentially warts in humans. And without the vaccine, most humans at some point during their lives will pick up HPV through contact with other humans. And the problem with it is that virus tends to inject or insert its DNA into our DNA in such a way is that in females it tends to induce cervical cancer. All right, so cancer of the cervix, which is a narrow part of the uterus where it kind of merges with the vaginal canal. For whatever reason, this HPV virus tends to induce cancer in the cervical region in humans, and possibly ovarian as well, cancer. So this vaccine was developed and designed to be used and administered to people at a point in time before they were likely to have picked up the virus from other humans, specifically through sexual contact. So the vaccine name is Gardasil. That's the name brand. And so Gardasil, to be most effective, has to be administered before someone becomes sexually active, because you get the HPV virus, this form of the HPV virus, through sexual contact. Doesn't really necessarily always cause any other symptoms of any kind, but it does greatly increase the chances of someone developing cervical cancer. So if you get the vaccine before you're sexually active, this prevents you from getting the virus ever. Okay, so of course there was a big controversy. All sorts of groups sort of took this and bent it or twisted it to mean that all of a sudden doctors were recommending that kids get a vaccine so they could start having sex when they were 12 years old, which was totally not the case. They were trying to get the vaccine out there in people before they were sexually active, whenever that is, so that whenever that does occur, they don't pick up the virus. All right, so it's very effective. It's actually, I think, now a series of 2 shots instead of 3. So most people, so you guys are in the generation where you probably would have gotten Gardasil as young adults. It used to be 3 shots. Now I think it's 2. So it's actually not even as painful as what it used to be, which is another benefit of that. So this actually, this idea of viruses inducing cancer, it's not just HPV. It's not just cervical cancer. There's a lot of evidence that various forms of herpes viruses could induce things like breast cancer, Hodgkin's lymphoma. There's really a lot of unknown as far as how likely or how often viruses are in inducing cancer in people. Something else that occurs, which is kind of interesting, and this sort of gets into a little bit of immunology as well, is that when a human cell is infected by a virus, at times it will produce what are called interferons. And interferons are essentially a way for an infected cell to warn other cells in the neighborhood, in the vicinity, that they're sick and that they might be in increased risk of releasing viruses in the near future. So other nearby cells can raise their defenses so that when the viruses are released from the infected cell, they have maybe a less or a lowered likelihood of getting into the other cells in the nearby vicinity. So that's actually something which is kind of interesting as well. And then we get into kind of discussion about how like cytotoxic T cells or T killer cells can kill off cells that are infected by virus. It's a really cool topic, but just sort of a little bit of intro there as we kind of wrap up this chapter. Smallpox was a disease that was historically, like I said before in earlier section, very important to humans. We still think there's a possibility of a human pandemic, even though smallpox has been completely controlled in humans. There's not been a case of smallpox since 1979 that was natural. The reasons for that is that it's very, very easily controlled with a vaccine. Vaccine does leave a fairly serious scar on the shoulder where it's typically administered, but otherwise very effective. The symptoms of smallpox, like you see in this bottom picture, are very recognizable. You don't really see any other disease that produces those pustules like smallpox does. So if somebody were to come into an ER covered in those, pustules, like chickenpox times 1,000, doctors would recognize it. Even though, modern day doctors, most of them have probably never seen a smallpox case in real life, they would know what it is because it's so distinctive. There is no inoculum reservoir in nature, so it's not in the water, it's not in the soil, it's not in the air, it's not in any other animals that we know about. So the benefit there is that if we control it in people, it's controlled. We don't have to worry about it being reintroduced to our species from other species. It also doesn't mutate very well, so that's another factor which makes it sort of a benefit to us, is that it doesn't change all that often. The vaccine, therefore, is very effective and will continue to be likely for the long term. With all that being said, there is still smallpox virus in the world, and most of it is kept in research facilities. It's studied, it is monitored, it's kept on lockdown, it's sort of high-level containment. It's important to study these things, but there's always a little bit of a risk that maybe some of the smallpox inoculum might escape one of those facilities and be reintroduced into the human population, which would be probably fairly catastrophic, at least for a little while until we got the vaccines rolled out again. Most people now don't even get vaccines against smallpox unless they're military and they're being deployed overseas because there's worry about it being used as a bioweapon. But otherwise, general civilian population in the U.S. does not get a smallpox vaccine. I never got one, and I was born in '78, so it's been a while since people have gotten smallpox vaccines large scale. But if it were to get out and get reintroduced to the human population, it would very likely do quite a bit of damage before it was controlled again. All right, so this is one of my favorite slides in this lecture. It's the last slide, but it's one where we start getting into some other things that are similar to viruses, but are actually smaller than viruses. So there are 2 examples here. There are viroids, which are small pieces of RNA that are not surrounded by anything. They can cause diseases, especially we think in plants. We don't really know of any viroids causing illness, say in humans or animals, but there are some that can cause damage to plants. And then there are the prions. And prions are most famous for causing what are called TSEs. So TSE stands for transmissible, meaning can be passed on, spongiform, which refers to what it does to the structure of the brain, the physical change that it does to the brain, and encephalopathy, which refers to a swelling of the brain. All right, so none of that sounds very good. Transmissible, spongiform, encephalopathy. The effect of this on organisms that get it is typically pretty catastrophic, right? So basically what these are caused by are prions. And a prion is an infectious protein, right? So back before the early 90s, people thought that only things as big as like a virus or bacteria could cause transmissible disease. Here we have non-living proteins that not only can cause disease, but they can also be passed on to other hosts and make them sick, which prior to 1990 or so, people didn't think was even a possibility, but it turns out they can do that. So these proteins, when they get into an animal body, cause other proteins around them to misfold, essentially denature. And when all those proteins just denature or break down or lose their shape, they cause proteins around them to break down and lose their shape, who cause proteins around them to break down and lose their shape, and so on. It's like a chain reaction where these proteins are just broken. As soon as one of them gets into the body, you start seeing many millions of them being broken as a result of that. So scrapie is what's happening to this sheep up here. Scrape is a condition that is neurodegenerative. So it causes a breakdown of the central nervous system. It's also chronic, which means it's sort of long-term until the animal dies as a result. It's transmissible, which we'll talk about here in a few minutes as well. But the effect of scrapie, the symptom of it, is that when a sheep has scrapie, it will literally go and find something to rub its body on because it feels like it's itchy. Right? Even though it's not really itchy, its brain is interpreting the sensation of itchiness from sensory cells in the skin. Even though there's no stimulus there in real life, the brain perceives a stimulus that's actually not there. So the sheep will find a gate or a fence post or the side of the barn. It will just scrape its body back and forth on that surface to alleviate the perceived itchiness, to the point where it actually rubs off all of its wool, its fur. It rubs big open sores in the skin, and it eventually does nothing else except rub its body on those fence posts or sides of barns or whatever, because the sensation is so overpowering. They just do that nonstop 24/7 until they starve to death and die as a result. In New Guinea, there's a tribe of people called the Fore people that anthropologists back, I think in the 70s or so, discovered. These people discovering from a condition called kuru. And so the Fore people in New Guinea are cannibalistic. When someone of authority in a tribe dies, they are consumed by the rest of the tribe as a sign of respect, which is part of their culture. And so it was found by these anthropologists who were studying these Fore people that after these cannibalism rituals, there would be large waves of women and children coming down with this condition called kuru by the local people, that essentially the people would just go absolutely insane. They would start acting very erratically, very strangely, very illogically, and nothing anybody said or did would change that. They were just acting completely insane out of their minds. Well, it turns out that the kuru prion is carried in the cerebral spinal fluid of the people that die from it. And the women and children in the tribe of the Fore people would consume the brain, the eyes, the spinal cord of the person that died, and for that reason they exposed themselves to large amounts of CSF, or cerebrospinal fluid, and therefore they picked up the prions from the person that had died. The men in the tribe, of course, consumed the meat because they were macho probably, and for that reason men were not exposed to these prions, and men never really got the kuru condition. It was only women and children. Right? So it takes exposure to the prion to actually pick up the condition, but once you do, it's a pretty much chronic progression on the way to death. The scary thing about prions is that, yeah, they're infectious proteins, but they're also not especially prone to denaturing on their own. So, for example, if you were to eat a sheep with scrapie or a cow with mad cow disease, you would be getting the prions, you'd be consuming the prions in the same form that they were when they were in the animal. Now, chances are those prions may not cause a lot of problems in the human body, but they can at times, as is the case with mad cow disease, cause issues with the human body and even cause death of humans. If someone consumes mad cow tainted beef or sheep, lamb, whatever, if they consume that, it contains those prions. It doesn't matter how well you cook it, doesn't matter what you do to it, you could immerse it in acid if you wanted to, and these prions are very, very stable. They do not break down, so consuming a cow that had mad cow disease is never safe. No matter how well you cook it, no matter what you do to the beef, it's always going to have that prion present, which could make you very, very sick. So in mad cow disease, it's a bovine spongiform encephalopathy, which is what BSE stands for. There's also a chronic wasting disease in all kinds of deer, so white-tailed deer, black-tailed deer, elk. All these animals are susceptible to chronic wasting disease, where the virus in these animals causes them to just basically stop eating. That's the main effect, is they just lose interest in eating and they waste away to nothing. They a lot of times will find some other behavior that they will pursue almost compulsively. There's stories of deer with chronic wasting disease just walking in circles in a field for days and days on end until eventually they just die and pass out due to exhaustion. Or I guess they pass out and then die as a result of exhaustion and dehydration probably as well, because they've just done nothing but walk for the past 7 days and they starve themselves to death. So very scary stuff. There's also a link between prions and Alzheimer's disease. Some of the beta amyloid proteins that are or have been implicated in causing Alzheimer's appear to have a prion-like behavior where they can, as soon as some of these prions begin to build up in the brain, they cause other proteins around them to become prions as well, which then causes kind of ever-magnifying neurological damage. So that's actually something which is still being studied currently, has been studied by faculty at Lakeland as well. Dr. Nussbaum has worked on that in his past. So really interesting stuff and really something we're learning a lot more about. So that's going to wrap up our chapter on viruses. I talked about it a little bit more than I usually do due to current events, but hopefully you found that one interesting. The next chapter we're going to get into is the one on prokaryotes, bacteria. So we're going to wrap up this section here, and I'll start up on that next one.